And we will now start our review. Okay, let's bring it up. Or I guess Sam should be starting us.
Yes, so let me go ahead and share my screen. Oh my goodness. Okay. All right, cool. So basically we are, yes, I will share the link. We have some questions for you guys, and like I don't think it'll necessarily take the whole time. So with that leftover time, we can go over exam questions and stuff if you guys want to talk about that. But let's just get into it. All right. So, first question is, what's the significance of using a primer? Like, why do we need it? What's the point? Oh, it says access to, oh, let me change that. Okay. It should be working now, hopefully. Meanwhile, does anybody want to tell me what the significance of using a primer is? Or I guess another way to ask is, why do we even need a primer? Oh, yes. Okay. So Joe said, yes, it provides the OH. So basically, DNA polymerase cannot add to, it can't just like synthesize on nothing. Like it has to have something to add the nucleotides to. And so using these primers, they provide an OH, a hydroxyl group, in order for the polymerase to actually add on to it, because polymerase, it has to have something to add on to, and it adds on to the OH. And so that's, yes, Casey, that's part of elongation. All right, next question. What direction does DNA replication take place? So 3' to 5' or 5' to 3'? Yes, it's 5' to 3'. And so this means that we read in the 3' to 5' direction, but then the nucleotides actually get added from 5 to 3. But it's kind of funky because we know that our DNA strands are anti-parallel. So like one runs in the 5' to 3' direction, and the other one runs in the 3' to 5' because we're anti-parallel. And so since we're only able to synthesize in the 5' to 3', what is a consequence of this? Exactly, Casey just have it. So we get Okazaki fragments because we have discontinuous elongation. So basically what happens is you, on the strand that's 3' to 5', like everything is good and it's great, and you continuously synthesize because you read 3' to 5' and then you synthesize 5' to 3'. So like that strand is good, but its complementary strand, the other template strand, that's running 5' to 3', which means you have to unzip it a bunch and then synthesize from the inside to the outside, and then you have to unzip it more and then start all over again. So basically you get a bunch of these little pieces of newly synthesized DNA that are not connected together. And so does anybody know which enzyme we use to connect the fragments? Yes. We use a ligase to connect those fragments. And so that's something that's kind of funky about synthesis, but the cell, like most things, found a way to get around it. All right. What could be a consequence of having a faulty DNA polymerase 1? Emphasis on the one. Yes. So do you want to emphasize or explain what DNA polymerase 1 does? Yes, so it can't fix the mistakes if it's faulty. So basically, DNA polymerase, your polymerases can carry out some proofreading activity. And so, yes, Casey, DNA polymerase 1 is the proofreading enzyme. And so it can act as an exonuclease, which basically just means that it can go back and remove nucleotides from our DNA strand that we're synthesizing. And so since it can do that, it has the ability to go back and fix any mistakes. But if it's faulty or if it's not working properly, then it can't proofread. And so it can't go back and go over the DNA and go, oh, okay, that nucleotide is wrong, so it shouldn't be there. And since it's unable to proofread, if you have mutations that arise, they don't get fixed and they just like are there. And so when you go and you try and replicate that DNA again, because it's been mutated, because you couldn't proofread it, like that mutation just keeps getting replicated over and over. And it's just not a good time, you know? So does anybody have questions about any of the first three questions?
I have a quick question. So
Mhm.
DNA polymerase, when you say that it removes nucleotides, that means it removes nucleotides that are basically like misplaced, right?
Yes.
Okay. And the nucleotides, that's like the bases.
Mm-hmm.
It's a very basic question, but I just want to make sure. Okay,
Yes.
thank you.
Yeah, no problem. Does anybody else have any questions about that? The content now that we're going over is less like math based. I'm sure you've noticed, and much more conceptual. So I don't know, it's just like a change of pace from what we've normally been doing. But if everybody is cool with that, then Garrett, do you want to take over? Yes, Joseph, it is a lot like cell bio. It's like cell bio, but more in depth.
All right, a lot. So replication. You and Sam just talked about it a lot. When is this even happening? During S phase, exactly. And all the time, double exactly. Great work, team. So just generally, DNA replication is always happening, and it has been happening literally every moment since you became like an independent organism, even before you were an independent organism when you were growing inside of your mother. Because, in order to grow and go through mitosis, in order to grow and go through mitosis, you need to like constantly create more DNA so that all of your new cells have DNA. And like Casey mentioned, specifically within the mitotic cell cycle, this happens during the S phase, which is the synthesis phase, where we're synthesizing or replicating new DNA. And how is DNA synthesized or replicated? In what manner? And yes, Casey, it does slow down with aging. That's not what I was going for, not the direction. But how is DNA, how do you go from one strand of DNA to another strand? Semi-conservative replication. Excellent. Very good. All right. Great work, team. So now that we have, now that we know when this is happening, let's like make sense of an image, a depiction of replication. So what is this picture that you're looking at? What are we trying to make sense of? That is a replication bubble. Excellent, excellent, excellent. So make sense of this replication bubble, if you please. The things that stand out to me when I first look at a replication bubble are these guys, or, oh, you can't see my pointer. 1 second. Yes. Okay. Now can you see my guy? Sick. Okay. What are these guys? They're the first things that stand out to me.
I cannot see your pointer.
Okay.
So I don't know what you're pointing at.
Thank you. No, I was not trying. What are these guys? Thank you for letting me know. Yes. These guys. I'm gonna not use yellow. These guys are gyrase. What does gyrase do? It stabilizes our DNA because, you know, DNA is like a really, 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 really long molecule and it's very thin. And so it can snap easily if it's untwisted too much, too quickly. And, you know, we're doing this really fast. So we need something to stabilize our molecule so that it doesn't get just shredded and obliterated as we go through our stuff very fast. Gyrase is not there all the time, only when you need to uncoil it so that you can build new complementary strands for replication. Good question. Very good question. What's next? What else jumps out at us? The blue things. What is the blue thing? And I hope you're not just like looking at the slides, because that would be not testing your powerful brains. You need to test your powerful brains. Probably the proteins that keep it open. That is a great guess, but it is not accurate. That is also a great guess, but not accurate. These blue thing guys, notice that, well, I guess they aren't labeled, so it's kind of hard. These blue guys are our primase enzyme. These are the enzymes that attach primers to our original template strands. So that, like Sam mentioned earlier, oops, how do you do that? So that's, oh my gosh, when Sam mentioned earlier that DNA or polymerase doesn't just like go, go, go. It needs something to like latch onto to start its replication in the first place. Oh, well, thank you for coming. We understand. But yeah, thank you for coming. Yeah, DNA polymerase needs something to attach to to start its replication in the first place. And primase does that by providing primers. It primes it. And so I kind of gave it away. But what are these green things that we see? Bingo. This and this are our primers. And these primers, because if we look at our, if we zoom in and look at the nucleotides, we have some sequences. Primase put these sequences here so that when our DNA polymerase comes along and it wants to attach complementary sequences, it has something to go off of. So what's up next? The orange is ligase. I would not say so, but that is a good guess. I don't think, just to be thorough, ligase is not pictured here. Yes, helicase, exactly. So we have some helicase. And what's helicase doing? Yes, it unwinds. Unwinds what? The double-stranded DNA, exactly. Helicase unwinds this helix. Very nice and easy to remember. Okay, so there's one last, or maybe two last components to this that I'd like us to mention. So what we're looking at is the replication bubble. Where does the replication, oh, very good, Casey. These red strands, wait, let me make sure I use your wording. Yes, these red strands are the new complementary or non-strands. There's one last element that I'd like us to discuss. So we have this replication bubble, like I mentioned. Where does it start? How does it get going? Is there maybe a specific point that it starts at? Maybe. Yes, the origin of replication. Right here and right here. So we have this very nicely in the center of our replication bubble, the origin. And what chemical or enzyme or molecule decides that this is the origin point? Indicator protein. You're probably right. It's called ORI-C. ORI like origin. Very cool. And so we kind of just labeled what's on this side of the replication bubble. But what we need to realize is that, and it's hard to see because this is a still image trying to display a moving process, is that from the origin point, this is going in both directions. This helicase here is unzipping this DNA, and this gyrase is stabilizing continually upstream that way, while this helicase here is unzipping this way, and this gyrase is going downstream this way. And we have this, okay, let's identify. out of these 4 strands of DNA, which are, or let me do that in a different color, sorry. Which are the leading and which are the lagging strands? Top is leading, bottom is lagging. You can't, the thing that's kind of the trick to this is that just because you have top and bottom doesn't mean that one is leading and one is lagging, because replication goes in both ways. So no matter which side you're on, top or bottom, there will be a leading strand on going in one direction and then a lagging strand going in another. Same with the top. Which direction is it going, Joseph? You're going to have to ask that question or answer that question. Oh, actually, I guess you can't really see it too well, but this actually, we do know. So we have this really long strand, and because it's not broken up into fragments, we can probably guess that it is the leading strand. And if the leading strand is going, or we know that the leading strand always is built in the direction, sorry for my handwriting, of the oops, get out of here. Go away. It goes in the strand of the replication fork. This is the fork right here, and then gyrase is actively moving the fork along your DNA. So the leading strand is always built in the direction of the fork, because we start at the origin and it is being built toward the replication fork that will continue to move that way. Similarly, on the opposite strand, we have, or on the anti-parallel strand, we have this strand that is being built toward this replication fork. So these two are leading. And because this one is leading, which direction, which side is 3 and which side is 5 prime? What's this one? You should be able to look at any picture of replication going on and be able to deduce every element of it. Oh, is that the question that I asked, Joseph? I meant to ask which side is 3 prime and which side is 5 prime of this leading strand? Yeah, sorry. Joseph says that 5 prime is on the right. What do we know about DNA replication? We know that DNA polymerase builds or replicates in or from the 5 to the 3 prime end. And because we know this is the origin, obviously starts at the origin, we know it's built from the 5 to 3, and it's built from the origin, the 5 prime, to the 3 prime. And if we have the 5 and 3 prime of our, I'm going to make this a lot less messy, sorry. And because we know that this is the 5 prime end and this is the 3 prime end of our complementary strand, what do we know about the direction of the template strand? It's opposite, exactly. It's anti-parallel. So if this is 5 prime and this is 3 prime, then this is 5 prime and this is 3 prime. Right? Very cool. And like I said, it's kind of hard to just look at a still picture and be able to tell what's going on. So what helps me is,
Oh, sorry.
oh, what's up? Do you have a
No,
question?
finish your thought. I do have a question.
Okay. This is just a little GIF that I found that's kind of like helps. So we have our 3 and 5 of our original. The red is our template strand, and we have this blue complementary strand being built. And we see that it is being built from the 5 to the 3' end, like we know DNA is replicated in. And so this is which strand? Well, I guess it's given away. This is the leading strand because it is being built toward the replication fork. Excuse me, and this is our lagging strand, obviously because it's being built in pieces known as Okazaki fragments, but also because it is being built away from, you see the arrow right here, and it's being built away from the replication fork. And would you like to ask your question now?
Yes, I was just going to ask how you determine that how you determine the leading and lagging strand.
Oh yeah, so we know that this is our origin, right? Everything starts at the origin. That's the meaning of the word. And we can see that we have 2 replication forks. I'm going to switch colors because that's not very evident. Purple. Origin, and then we have our 2 forks. We know that everything starts at the origin. So when we see a strand that is being built toward the fork, we know that's our leading strand. And similarly, we have something starting at the origin going toward the fork. So that's also a leading strand. But DNA is also being replicated in this way away from the fork. Oh, actually, no, that's not true. DNA is being replicated this way because, you know, it's only replicated from 5 to 3 prime. Did I answer your question? Let's see. Oh, you didn't type it in the chat. Yeah, so I know that it's the leading strand because it starts at the origin and it is going toward this replication fork here.
Yeah, that makes sense. So basically, like if we can identify the fork and then look at the origin site, if it's the strand is going from the origin site to the fork, we know it's the leading strand, and then it's the lagging strand if it comes after the fork towards the origin.
Exactly. It's being built from the fork toward the origin because it's only built from the 5 to 3 prime end. And we know that our complementary strand has its 5 prime end on the, I guess you can't use left and right, so I'm not going to try to confuse you. But yeah, just like you said.
Okay, that makes sense. Thanks.
You're welcome. And another way to visualize it is this very cool video, if you'd like to watch. Oh God, what happened? Oh no, stop that. Don't wait. I think I accidentally pressed fast forward. I know, this is so cool. So we have our DNA obviously being coiled around what? What do we think those purple guys are? Histones, very good. Well, let me increase the quality so you guys can kind of see it better. Oh, it is. Shoot. Sorry. Is it still super laggy? Okay. Well, you guys can watch it on your own, but this is kind of what I wanted to show, that like this is the real-time speed of your genes replicating. I highly encourage you to click on this link and watch it. It's amazing. And so another thing that I wanted to talk about is we've said it a few times. It is called the Okazaki fragment. It's the special name that we have for these lagging strands of DNA that, because they're replicated in the 5' to 3' direction, they start upstream of the origin at the fork and then are replicated toward the origin. And the name Okazaki fragment comes from the scientist who discovered it. And this person, I think her name was, actually, I don't want to misremember. It starts with a T. I think it's like Tenuko. Tsuneko. Okay. So there is a scientist, a geneticist named Tsuneko Okazaki, and she and her husband were the pioneers of DNA replication genetics. And because she was a woman, her husband was given credit for the discoveries rather than her, despite she being the primary author and definitely the more profound geneticist. So here's a little podcast I've linked if you want to give it a listen. Not just the wife. Okay, so moving on. Please order the following types of cells. Yeah, please order the following types of cells. And the ones with more telomerase working on their DNA should come before those with less telomerase working on their DNA. I'll give you a second. Oh, and a hexactinellid is a glass sponge. It's just a kind of sponge that uses silicates in its pinacoderm to ward off predators so that it doesn't get eaten, because you don't want a mouthful of glass. So I could edit that to make it more palatable. I should also note that glass sponges are known to live for many thousands of years. I think up to 10,000 is the longest that's been known. Exactly, Casey. That's what I was trying to get at, is that some organisms are just obviously they utilize more telomerase than others. And that's why they don't age, most likely. And Joseph has it perfectly right. Excellently done. The answer is that cancer cells utilize the most telomerase, and then a human's germ cells. The reason cancer uses the most telomerase is obviously because they're mutants, they're doing stuff and they aren't following the normal instructions. They want to perpetuate themselves. And that's why they grow so much and why they're kind of immortal and why they're so hard to get rid of, is that they upregulate telomerase within their cells so that their DNA is constantly being repaired. It's not being degraded. And so they can keep replicating and keep acquiring mutations without having to worry about the end replication problem. And then germ cells, humans' germ cells, as opposed to humans' bone cells, humans' germ cells, they also upregulate telomerase, because your germ cells are the ones who go through meiosis and who create your gametes. And so if say you're 20, well, I'm 22 years old. Say I wanted to have a child. And if I were to have a child and my germ cells didn't upregulate telomerase, then the 20 years or 22 years that I've been alive, my telomeres have probably been shortening, because of the end replication problem. And so like I would be, if my germ cells weren't utilizing telomerase to like repair my telomeres, then I would be passing on to my child like 22-year-old DNA that's been shortened throughout my 22 years. And so what my germ cells do instead is that they upregulate telomerase so that my gamete can have like fresh new DNA. So it's not like pre-shortened. So my child isn't born as a 22-year-old. And then, uh, sponges, they live for tens of thousands of years. So they probably utilize telomerase to limit the amount of aging that happens within their cells. And then the same method or mindset for tortoises cells, because tortoises are known to live for hundreds of years, and then human cells. And mammals aren't very long lived. So yeah, very cool. Did I explain the germ cell business well enough? I could try again. Okay. I'm not sure how to answer your question. All our cells age. We're constantly, yeah. That's why I was a bit apprehensive, because like there is obviously for some individuals, more so female mammals, I think, than male mammals, that actually, I'm not going to comment on that because I really don't know. I think complication with bearing children doesn't come down so much to your meiosis as it does to like just your ability for your body to grow a child rather than to make the gamete that goes into a child. So maybe not actually. But again, I'm not like perfectly sure. So that was all that Sam and I had for you. Do you have any other questions? Okay, MCM. Let's have a look. Oh, yes. 1 second. Oh, yes. Well, thank you for coming, Joseph. Yeah, this one was probably going to be short. So we're going to finish up soon. Let's talk about the MCM, though. Do you have any ideas about that, Sam?
Sorry, Gary, can you say that again?
Oh, do you have anything you want to say about the MCM?
No.
No problem. um, where is, okay, here it is. Sorry, I'm just pulling up the slides. Okay. So the background is that we want to make sure during replication that replication only happens once. Like we don't want to have within a single replication bubble, we don't want like multiple leading strands. We want one leading strand protruding from the origin toward the constantly moving replication fork. So what we're going to do is have an MCM, which is a mini chromosome maintenance factor that kind of like, it's a one-time permission kind of thing. Like it grants permission for replication to begin one time at a specific origin. And that will be our one replication bubble. So MCM is kind of like the, I guess, secret password, for lack of a better term. It's the secret password that can be used one time to start replication at a certain point. Or I guess to use the terminology, it's a license. Because you know how you have to like buy, for some reason, schools require you to buy licenses to homework programs for classes that you're already paying a lot of money for. So kind of like you need to buy a license to get access to the homework. The replication process needs a license to access the DNA within, or I get the DNA within DNA. I'm not explaining this well, but yeah, MCM like has the permission for replication to start. And once it gives that permission, replication can't happen anymore at that site.
Okay, that makes sense. It sounds like it's kind of like the gatekeeper almost. Like
Mhm.
here's the origin of replication, and it allows replication to occur at that origin with just one strand.
Yeah. I'll give you a second to think if you have any more questions.
I'm good to go. Thank you guys both for having these reviews. It was especially helpful for this exam. So thank you.
Oh yeah, no problem. Thank you so much for always coming to them.
Yeah, of course. Always be. I'm glad that like I don't have a schedule conflict. So fortunate. Anyway, I'll see you guys next week and have a great weekend.
Awesome.
No
Thank
problem.
you.
Thank you. You too.
Thanks. See you guys.
And Michelle or Caroline, did you guys have a question?
Nope, I'm good too. Thank you guys so much.
Awesome. You're very
problem.
welcome. Okay, well then I think we can separate for now.
All right.